This talk is going to be about protein fitting, an introduction to protein fitting. I am going to talk more from a practical point of view. How can we use Chimera for actually do protein fitting? Please feel free to stop me at any point and ask any question. Okay, so what is protein fitting? I think now it's quite clear for everyone what protein fitting is. But maybe if we go back 5-10 years ago, sometimes fitting and docking were used for the word fitting and docking were used for both processes. But let's explain this with this example. Here we have a 16 Armstrong resolution map. This is what most probably we will get after preparing our samples, going to the microscope, analyzing all the images, and finally to the three-dimensional reconstruction. The problem is that from a molecular point of view, this object doesn't contain too much information about the molecular mechanisms that are behind the function of this complex. So, if we want to know the function and explain the molecular mechanism, what we need is the three-dimensional structure. However, large assemblies are quite difficult to crystallize, and it's quite difficult to solve their structure. And it's the same for example, other techniques as NMR. NMR cannot deal with large complexes. So the problem is, how can we obtain the structure of this complex? So, what is easier to get is the structure of the subunits. They are smaller, probably they are easy to crystallize, or maybe with NMR we have other tools for homology modeling. And this is what is protein fitting, is the quaternary structure modeling of a complex using the structure of the single unit and the volume of the whole complex. And this is the difference with docking. In docking we don't have this information, the information of volume. So let's see with an example why we need the structure of the complex to understand the molecular mechanism. So this is a type 1 enzyme that is involved in DNA methylation and the transmission of genetic mobile elements. And with only this thing, with only this object, it's quite difficult. For example, imagine that I want to design an antibody that binds to the DNA binding site region and blocks the interaction with the DNA. So, with only this information, it's going to be almost impossible. But if I am able to find the structures of the units of this complex and locate it within my volume, I will be able to analyze the different biochemical features of this structure, and I might have the possibility to find where the DNA is binding to this complex. So, well, just summarizing, why protein fitting is necessary? Because or we have the structure of our protein, or otherwise it's going to be quite difficult to understand the function, explain the molecular mechanism. The problem is that the structure of large complexes is quite difficult to solve by other techniques as X-ray crystallography, NMR. So what we can do is to solve a low resolution structure by, for example, cryo-electron microscopy, and then try to use algorithms to fit the structures of the single units within the volume. So, okay, protein fitting is not the only thing that we can do. We have different scenarios depending on the resolution of our volume. We have other tools that can be used. So, between 10 and 30 angstroms resolution, we can observe the different subunits of our complex. Between 6 and 10, we can locate the secondary structure. Alpha helices are easier to see in the volume. We need volumes around 18 angstrom resolution. Beta sheets are a bit more difficult. When we have a high resolution in the context of EM, then the backbone is traceable. So we have tools to do that. So if we don't have, here, of course that here you can still using fitting, but here we have other tools that can help to trace the C alpha backbone. And for maps that are with a resolution higher, lower than 4 angstroms, side chains can be resolved. So here we in fact can model the full structure. Okay, so as we say, 30 angstroms. The only possibility that we have to finally model the quaternary structure is by means of protein fitting. 16 angstrom, we can predict secondary structure. However, secondary structure is not the whole structure. So this scenario is also good for protein fitting, and this information is going to be very constrained. So probably I'm going to get a good solution. And okay, under 6 angstrom resolution, of course that you can still use in fitting, but it's better to use program for trace the backbone of the complex. I am going to focus this talk on protein fitting. So I am not going to explain methods for locating the secondary structure or tracing the backbone. So why fitting methods are important? It seems, well, and of course that every year we are getting better resolution. In this chart of here, you can see the higher resolution structure submit in EMDB every year. So it seems like now we are getting a really good structure, and for angstroms, we can do a lot of things. We don't really need for those structures go to atomic structures and use all these algorithms. However, when we look to the whole database, the situation is quite different, because the structures in fact that are lower than 5 angstroms are only 3% of the whole database. So for all these guys here, we need to do something, because we need, if we want to have the atomic structure, we need to have some methods to fit the structure there. Okay, so now I'm not going to talk in details about the algorithm. I'm going to explain, well, just briefly an overview. All of them are very well documented, and if you want to talk later about them, there is no problem. Okay, so I have focused on Chimera. Well, probably all of you know what is Chimera, but just in case, it's a program for structural visualization and analysis of molecules. I think Chimera is probably the most popular viewer in the electron microscopy context. And well, I think it's a very user-friendly software. It has a large community of users and developers, so it's quite easy to get any help. Just go to the mailing list of Chimera and you will get an answer to any problem that you have. Also, there are a lot of developers programming extension for Chimera. They are very easy to install. The only thing that you have to do is to copy a folder in the extension path of Chimera, so it's quite easy. It's available on all the operating systems, and it can read probably the most important formats for electron microscopy and protein structure. Okay, this multifit is one of the algorithms that I'm going to show you. It is included in Chimera, in the standard distribution. Before was a plugin, but now there is no need to external, to make an external installation. Well, it's a program to perform multi-body fitting. It's also available as a web application, so if you don't want to use Chimera, you can go to this web page and it's almost everything the same. And well, what you need to run this program is the atomic structure of all the subunits and the volume of the three-dimensional of your complex. Okay, so what we have to do is open our volumes and the structure. Well, just in case someone wants to repeat this experiment, here you have this data. Here you have the data of the volume and these structures. So what we have to do is first open everything with Chimera, and to open the window of multi-fit, go to Tools, Volume Data, and multi-fit. And we will get something, a window like this. Here we have two options. If our complex has symmetry, we only need one copy of the structure. If we don't have symmetry, we need all the structure for all the subunits. So here is just the number of copies of the symmetric unit. And well, here we have two options. If we have opened different maps, we can choose actually. Pero está enchufado, ¿no? It is plugged, no? Isn't it? Yes. It was a bit. Okay, and also the same for the atomic structure. If you have different structures, you can choose the one that is your asymmetric unit. Okay, so in our case, we are going to choose no symmetry. So here we can select the chains that we want to fit in our map. So we don't have to fit everything if we don't want. Well, we will see this. And we can choose here the map. And the only thing, I will explain later the options, but first we have to just view the algorithm. And well, we just click on the fit button and we get something like this. These are the results. So here we have it says correlation. This is not a real correlation. This is the score of that multi-fit is given to each solution. The lower is this value, the better is supposed to be the solution. However, this is a mathematical model. Maybe the best solution is not the closer to the native structure. So what you have to do is always check some of the structure and see the one that you believe the most. Okay, so by clicking one of these solutions, the model panel is going to be updated and the new change is going to be added in this panel and also in the viewer. So this is what we got. Here we have the solution in yellow color. So what I have done, here I have compared the solution with the native structure. In gray we can see the native structure obtained by X-ray crystallography, and I have colored instead of yellow with the respective color of the subunits. This figure maybe is a bit messy, but if we compare chain by chain, we can see that multi-fit is producing a very nice result for this case. Okay, these are different solutions. So for example, this is the best solution compared with another one. I don't remember which one was. But what we can see here is both solutions are agreed in these three units, but the fourth subunits are different. No? So which one is better? Okay, you can choose the one that has the best score, or you can choose another one because you know some experimental information that can use to filter the solution. And here, for example, we can see a very different conformation. So this is the correct one or the best solution, and in this other solution, multifit has put this subunit in this part of here. So multifit doesn't know the location of each subunit. It only tries to maximize a function. So okay, here is the same. The subunit that was supposed to be here in this solution is there. So what you have to do is always check the solution, and it's always better to choose the one you think is closer to a native structure than use the best score given by the method. Okay, so now let's explain a bit the algorithm so we can know what the different parameters are. So the first thing that multifit is doing is generating a graph. It's called the anchor graph. This graph, the nodes of these graphs are related with the region of the map. So this is important because here we can see that the graph have 4 nodes. These nodes are because I have told multifit to fit 4 structures. So for multifit, it's quite important to know the exact number of structures in our complex. So let's say that the program is not developed to perform partial fitting. So if instead of 4 I choose 3 structures, I will have a 3 nodes graph. How is this graph calculated? Okay, so I think Jose Millet talk a bit about the k-means algorithm. And well, this is in fact what is using multifit, is clusterizing these 3-dimensional volumes, these 3-dimensional points, using the k-means algorithm. Here we have an example in 2 dimensions of what k-means algorithm is doing. Okay, so let's say that I have this collection of points and I want to split them, cluster them in 3 different sets. So what the algorithm is doing is randomly I choose 3 initial points, whatever. Then what I'm doing is I mark with a color each one of these initial points. This color of here is green, this blue, and this here red. So all points that are closer to these initial points are going to be green. The same for that point. So for example, these points here are closer to these points than these or these. Then these points here are blue. Then I split our initial set in 3 sets depending on the distance. So each point is going to be the same color of the closest point, initial point selected. Once I have this cluster here, what I do is calculate the geometric center of all these points. That is this new point. This point, maybe it is not going to be one of my initial points. It can be just the geometric center. And I do the same for the blue points. This is the geometric center of the blue points. This is the geometric center of the red points. And now I can do again the same. I color the points depending on what of the initial points are closer. So I repeat this process until I have no change. So finally, at the end, when I don't have any change in the iterations, I get 3 different sets. So this is what the algorithm has done. Multifit is exactly the same. Okay, this is not real. I just erased the volume so I can show a bit, but the principle is the same. These points of my volume are closer to this point here, and this point of my volume are closer to this point here. And the edges of the graph, yeah, it's because here we have a discrete set of points, but the volume, these points are connected. So some points of here will be neighbor of some points of this region. So this is the reason why I draw an edge between these 2 points. Okay, so the edges represent connected region between my cluster, in my cluster. So what is the algorithm doing? The algorithm is trying all possible combination. It takes one of the subunits, put it in the center on one of the geometric center, and tries to maximize this score function. We will talk about what these terms mean by rotating and translating the subunit. So this guy here is here, but also we'll try to put this guy here, 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 and it will put different subunits in different points. So what these terms mean? The first term here is related with the graph nodes. Okay, so what the algorithm is doing is transforming the structure in a density map, and then is calculating this term of here is the correlation between the density map generated from the structure and the density map that we provided to the algorithm. Here is also a correlation, but not of the density map, of the surface of the density maps. And this last term is trying to evaluate the edges, how well fit these two structures. It takes into account the atomic structure, not the density maps of these two guys. What it's doing is calculating distances between atoms and finally produce a score depending on if I have a lot of atomic clashes, if the distances between atoms has sense in terms of protein-protein interaction. Okay, so it's amazing to see how close to the native structure is this fitting using a function that has anything to do with molecular dynamics. Well, a bit disturbed, but yeah, because in fact, after selecting the best function, the best conformation, using this expression, multifit is performing a docking between the different subunits. Is using patchdog to finally get the structure between the different proteins in my complex. So what it's doing is selecting by default the 10 best, the 10 best scored structures and applying docking. And these new structures obtained by docking are incorporated to my potential results and also evaluated with this function again. So finally, what we are seeing here is probably the best structure after the docking process. So for that reason, multifit can produce very close results, or at least results that has a lot of sense from a physical point of view. Okay, so when I click to options, I have these parameters here. First of all, fit globally. Fit globally is related with how multifit is moving the structures and rotating them to find the best scored conformation. So by default, the shift is 15 angstroms. So multifit will try this position, move 15 angstrom here, will try this position, also move here, will try, will again evaluate the conformation. If I click here, fit globally, multifit is going to use the distances between voxels in my map. Then it is going to take longer, the whole process, but also produce better solutions. Here, well, the number of outputs that we want to keep. This is the resolution. We have said that to compare the electron microscopy map with the structure is generating a density map for each subunit. So this is the resolution that we want to for the map that is coming using this structure. So this number should be the same that you have in your map. The sampling angle is related again with the option of fit globally. If we don't use this option, this argument of here has nothing. It doesn't affect anything. But by default, multi-fit is shifting or rotating the structures 30 degrees. So if we click here, we can change this number and use, for example, 1 degree. So it is going to try a large set of conformations. Okay, so if use it by default is quite fast, it's a very fast method. The data doesn't require any special treatment, so just the structures, the PDB structures and the volume and everything, and by default options is working quite good. Because it is using a docking step, multi-fit produce quite consistent modes. Of course, consistent from patch dot point of view. That is the docking method used. Of course, that the biggest limitation of multi-fit that we need all these structures of my complex. If one structure is missed, then we are not going to be able to use it straightforward. What we would have to do is to know where the structure that I don't know where should be my map and delete this region of my map. Okay, another tool that you can find in Chimera also is Segura. In this case, the method is performing a rigid body, not multi-body. So we will explain the difference. Okay, it can be executed from Chimera's desktop application. Here it should be the link to the Segura webpage. Well, it requires volume segmentation. So first of all, with Segura, we will have to split our volume in different regions. This is what multi-fit was doing automatically with the anchor graph. But in case of Segura, we will have to do it manually. Of course, that Chimera provides a tool. Argon is part of the Segura package to do this. Okay, and in this case, of course, that we can run partial fitting. So, and we say the first thing that we need to do is to divide this volume into smaller regions. So, okay, so I forgot to take another slide. Okay, so I am using the same multi-fit. But go to Tools, Volume Data, and you have here the option Segment Map. And when you click here, you will see a dialog windows like this, where the only thing that you can choose, well, we will talk again about the options, but the only thing that you choose initially is the map where you want to segment. Okay, so by clicking segment, we will get something like this. Segura thinks that we have two parts of this map, this region and this other region. Of course, that we need to fit for structures here, so this is not enough for us. But as you can see, here we have two options, group and group. So in fact, we can divide these two pieces in smaller pieces. But how does it work, this method? Okay, so let's go to one dimension instead of 3 dimension. Imagine that instead of these are now my voxels, and this is the density function of my map. So what it's doing, Segura, is sorting all the density values from the highest value to the lowest value. And I select the highest value. Okay, so this point of here is going to define my first region. So I go to the next value, which are my next highest density value is this here and this here. Okay, from this voxel and this voxel. What I check is this guy neighbor of another region? So this guy, no, it is not a neighbor, no, because we have this voxel here, this here, so they are not a neighbor. These two guys are neither a neighbor between them. So this is going to define another segment, and this is another segment. So okay, I decrease again the threshold of the density threshold. So now I have these 3 points. Okay, this point is neighbor of this voxel here. So as they are neighbor, I assign the same segment that was this other point. Also the same for this, and so on, until all the points are classified. What is the problem of that? That this method is producing a lot of regions, no? So what Segura is doing is merging them. And how are they merged? What Segura is doing, okay, let's smooth our signal, our density function, and I'm going to apply the same process to the signal that we have smooth, and I will obtain something like this. Okay, so now instead of 1, 2, 3 regions, I get just 2 regions. Okay, so some of this region has been merged, no? So what I'm going to look is to the maximum, to the local maximum in each segment, and check it if they are merged in this case of here. So for example, this point of here now is this. Okay, so this is only a set. So I mean, nothing else is contained in this set. So okay, this is going to still one region. But this guy of here that were maximum in the respective set, here now are in the same set. So what Segura is doing is merging these 2 different regions. So this is what we can do, grouping and ungrouping. Is going down until we think that the division is okay. So for example, if I now choose this segment of here and I ungroup them, I will obtain something like this. No? That, okay, this has more sense. I have to fit here 3 subunits and 4 subunits, and at least now I have 3 regions. Okay, so to fit one structure, we have to. Okay, no, this is a good option for the segmentation. So, smoothest steps. This is related with this step. So, how many times I want to smooth my signal, my initial density, and merge all the generated regions. This is the distance to consider 2 voxels neighbors. I can say, okay, I want to consider 2 voxels neighbors if they are closer of 2 voxels, 3 voxels. No, in the example by default is 1 voxel, but we can change algo this this number. Okay, so keep only region having at least one voxel. It has sense. I don't want. We can also choose if one region has less than 5 voxels. I don't want to consider it. Okay, so this is a way to stop the stop grouping. I don't remember what this means. Stop grouping if the number of regions. Okay, sorry, I don't really. You can look at help now. I don't really know what this means. Okay, display at most 2,000 regions. And surface granularity, sorry, I don't remember this option either now.
So that stop grouping, if number of regions is less than or equal to parameter, would let you set the least number of regions that you would want.
Okay.
So you can put 4 in there and you'll get 4 out.
Ah, okay, I see, okay. Yeah, well, okay. So now if we go to tools, volume, data, and fit to segments, we can show the window for a Segura. So here what we have to choose is first one of the regions. The regions are selected on the viewer. We have to control and left click to select the region where I want to fit the structure. So here I have to, once I have selected the region where I want to fit the structure, I have to select the structure that I want to fit to this region. And okay, we will talk about the options, but with the fit button, I will perform the method, and well, in that case, we also get a very nice result. Here we have in red color the result of the fitting, and in gray the native structure. So it was quite close to the native structure. So what options do we have here? Well, these treat all submodels as one structure. It is because in your PDB, if you have more than one model, you can ask Segura to consider all these models as the same structure. Density map resolution and grid spacing. This is the same because Segura is also transforming the structures into density maps, and this number should be the same that your volume. You can also combine different regions to group and perform fitting. So you can select 2 regions and ask Segura to perform fitting into selected regions. This is the alignment method. This is the score that will be used to align the generated density map with our map. So we have two methods. One is align the principal axis. So it is what is going to try is to find the principal components and align these components. This is a search in the three-dimensional space, the same that was doing multifit, just translating and rotating your structure until it maximizes the correlation. Mask map with region to prevent large drift. Yes, this is to consider. So if you mark this option, your volume is going to be considered 0 outside the segment that you have selected. This is to force Segura to fit the structure in the region. Use Laplacian filter. It will add a component that is the correlation between the surfaces, not the correlation of the density map, just also the surfaces. Optimize fits is going to do a second search, but it will have a better resolution. So the angles and the translation of the subunit is going to be smoother. Okay, we can cluster solutions if they are very close, depending on the sorry, I can't remember now this option either. I'm sorry. Okay, so and this is the number of solutions that we want to consider. So and finally, this is if we are using symmetry. No, it detects, it scores the atomic clashes between the different position of the structure. Okay, so we have selected one by one the different region and fit the structure, the corresponding subunit. Okay, so in this case, it didn't work so well, but what is important is that in this algorithm, we have to choose the subunit in the region that we think that the structure will fit. No? So in multi-fit, this was done everything automatically. This is a driven process. Okay, so, but what is, okay, so conclusion about Segura. It's a driven process, so the method, it performs rigid body fitting. We have to go subunit by subunit, and we have to decide where to fit each subunit. First of all, we have to segment the volume, but the tool is provided in Chimera. And well, in that case, as we have segmented the volume, we can perform partial fitting. Okay, so other tools that are useful when we are doing fitting. Sometimes we will not find the structure in the PDB. So to get the coordinates, we can also use homology modeling. Okay, these are some very useful web servers that one can use to model the atomic structures. Okay, and also we can use functional binding site prediction. So, for example, we have shown a case at the beginning where a complex was interacting with DNA. So we have servers to predict binding site region with DNA, protein-protein interaction, peptide interaction. No, we have these three web applications. So, for example, this is the case in the first example that I showed. Here, what we have done is to send the structure of the complex to the web application, and it has predicted what is the most likely region where the DNA can bind. And in this case, the prediction was quite good. Okay, so this is the work that we are developing in our group. What we have seen is that all these fitting methods, they are quite related with molecular dynamics. So we have seen some of them are using docking. We are searching for correlations between the structures and the electron and the maps. And what we are trying to do is to have a new perspective where the user could fit the structures by hand and then have some kind of a score. So how good are raw fitting that I can do at some point. So, okay, so imagine that we have this potential fitting. The first thing that we do is generate the protein-protein interaction graph. So what we can see here is each node represents one chain in our complex, and the edges are the interaction between the chains. Okay, now what we can do is exactly the same idea, but at domain level. We can decompose, we can calculate what domains are in my chains, and also the same. Let's see what contacts do I have in my complex between all these complexes. So, okay, so for example, here I have two chains, the red chain and the green chain. This is the same representation, but with different colors. I have represented a different domain in my chains. Okay, so here what I can see is this complex, this domain interacting with these domains in the other chain. So here you can see much better. One domain is interacting with these three domains. So now we have a graph where instead of protein-protein interaction, I have a lot of domain-domain interactions. And what I want to do is to score these domain-domain interactions. And to do that, I am going to use I want to score the interaction between A and B using the interactions between the domains in A and domains in B. So if A has these domains and B has these other domains, I want to finally compute a score based on the scores of the interaction between these domains. So how can I do that? Okay. The idea is, let's go to protein-protein interaction databases, take all the interaction pairs, and we can, for example, calculate the frequency that two domains appears in these protein-protein interactions. And of course that we have more sophisticated methods. We can use expectation maximization to finally have a better score. So once I have a score, all the domains interactions, I have these scores and I can calculate now the protein-protein interaction. So the idea is to score the protein-protein interaction in my complex using this information. And this information, you don't need a very nice model. You don't need a really good model from a physical point of view. So if I put the structures more or less where I think that they should be, no, I can still use this evaluation method. Okay, so thank you very much for your attention.